Welcome back to another YouTube video. Today, I will be reviewing the CSEC Biology exam topics for 2022. I'm Miss Disha, and I head the science block here at Disha's Online Academy. So guys, let's get right into it because I know in the next couple of months, I'll be birthing some students with straight grade ones, especially for biology. All right, guys, the first topic is growth. What are you going to do when studying for this topic? Well, you want to familiarize yourself with what is growth, and a common term tied to growth is development. So you want to go to the exam knowing what these terms mean. So for growth, growth is an increase in size and weight. Development is a process by which organisms become more complex as they grow. Growth and development involves three common processes: cell division, cell expansion, and cell differentiation. For cell division, this is actually the cell dividing, right? So it involves the parent cells dividing into daughter cells. A common name for that is mitosis. Then you also have cell expansion, right? Which we know is the increase in the size of a cell. And that can be as a result of osmosis, water or solutes going into a cell. Then cell differentiation is a common term, right? This is the specialization of cells to perform certain functions. So for plants, the very first instance of growth is seen in germination. And we're going to talk about germination in the next slide because it's an actual requirement for the examination. After germination, now the plants undergo two types of growth. In their younger days, they undergo primary growth. And then as they grow into big trees, they undergo secondary growth. So remember now, the animal kingdom is diverse, right? So you can look at growth in insects, arthropods. They show a form of growth when they undergo molting, right, or metamorphosis. Then now as you go to the higher animals like humans or other mammals, their cells increase. As a result of that, their tissues enlarge, their organs grow, and the whole organism therefore grow. And you can look at that from a baby to an adult. How do you measure growth in plants? How do you measure growth in animals? And look at stuff like taking the wet mass or the dry mass of an organism. In plants, you can take the length or the height. The second topic that you need to know for your examination is germination. And as a CSEC biology student, you must know the definition for germination, right? We know that germination occurs where the seed, right? The site of germination is the seed. And it is the process by which we have a dormant seed is going to be introduced to certain growing conditions. And then when that seed begins to sprout and grow into a seedling, that is called germination. And you must know the stages of seed germination. They can ask you that. For a seed to germinate successfully, they need the optimum temperature. Of course, you must have water and you must have oxygen. You do have certain conditions that affect the growth of a seedling, right? Some plants, they respond more to light. Some plants, they respond more to a certain pH. To master the concept of germination, you want to familiarize yourself with the types of germination. There are two types. There's epigeel and there's hypogeel. Epigeel means the cotyledons or the seed leaves, they come above the soil surface, right? So you want to know an example of a plant that undergoes epigeel germination. Hypogeel germination, in which the cotyledons remain below the soil surface. Before we end the germination, I want to see everybody going to the examination knowing how to draw a seed, how to illustrate the process of germination. Know your terms because they might ask you for annotations. So remember your germination vocabulary: radicle, plumule, cotyledons, epicots, tester, all of those beautiful terms. This is topic 3, nutrition. What is nutrition, students? How we eat food, how we take in food. I can just imagine some of your answers. Nutrition is the mode of taking food by an organism and utilizing the food for their growth and development. There are 2 modes of nutrition. There's heterotrophic and autotrophic modes. Let's start with autotrophic mode. So in this mode of nutrition, food is synthesized by organisms called autotrophs. So they make their own food, and they make their own food during a process called photosynthesis. Don't worry, we're going to talk about photosynthesis a little bit later, right? So how they do this? They obtain carbon dioxide and water and fix those into sugars and liberates oxygen, which we can use it for respiration. How is this mode of nutrition important? It is important because the autotrophs, they provide the food for other organisms in the environment to consume. Without them, we would have no food because they're the only organisms that can utilize light's energy to create sugars. The second mode of nutrition is called heterotrophic mode, right? So unlike the autotrophs, which can make their own food, the heterotrophs, they rely on other organisms for food. Moving on to the most anticipated topic, photosynthesis. Super easy. Once you know the site, requirements, conditions, equations, factors that affects its rate, you are good for the examination. What is photosynthesis? Photosynthesis is the process by which plants, green plants, use energy from the sun to produce sugars. Photosynthesis occurs in the leaves of green plants, specifically in the organelle chloroplast. And the chloroplast contains a pigment called chlorophyll, and it is the chlorophyll that is involved in the harnessing of the energy from the sun to fuel this process of photosynthesis. Before we get into the requirements, conditions, and so forth, the topic photosynthesis, you must know the structure of a leaf and the structure of the chloroplast. They might ask you to construct diagrams of these 2 structures. There's a thing that students often confuse themselves with. They don't know the difference between requirements and conditions. They're not the same thing, right? When it says requirements, we're talking about the raw materials, what is necessary for the actual process of photosynthesis. And then we'll talk about conditions, we're talking about the environment, what fosters the process, right? As it relates to requirements, you must have carbon dioxide. That's the gas that's going to be used to synthesize glucose in the light-independent reactions. You must have water. Water is going to provide the electrons to drive the process of photosynthesis. And then as it relates to conditions, you must have an optimum temperature for the enzymes that fosters the process of photosynthesis to work, and you must have light. Of course, what is going to the examination without knowing the equations for photosynthesis? You want to know the word equation firstly. Carbon dioxide plus water, you write your arrow, and then you have glucose plus oxygen. And on top of the arrow, ensure you write light, energy, and chlorophyll. Light intensity, carbon dioxide concentration, temperature, those 3 are what we call limiting factors. And we know that a limiting factor, that is something that limits the rate or the growth of something. As the light intensity increases, the rate of photosynthesis is going to increase, right? Until we have that area on the graph where photosynthesis stops. Similarly, as you increase in carbon dioxide concentration, that's going to increase the rate of photosynthesis. The carbon dioxide and the light intensity graphs, they are similar, but for the temperature graph, you're going to know its temperature is talking about. It looks like a bell where you have an increase in photosynthesis rate as you increase in the temperature, but it's going to stop at the optimum temperature and decreases until it stops. Moving on to the best topic in biology, cell biology. We know that the cell is the structural and functional unit of organisms. A high yield strategy going into the examination is you must know the different organelles found in plant and animal cells. There's the cell wall, and we know that this structure is only found in plant cells. The cell wall is a rigid structure made up of cellulose, necessary for providing support and shape to the plant cell. Next, we have a universal structure here called the cell membrane. It is found both in plant and animal cells. Unlike the cell wall that is made up of cellulose, the cell membrane is made up of phospholipids. And if you decide to do cake biology, and I hope you sign up for my class, then you learn more about that. The cell membrane controls what enters and leaves the cell. Next, we have the brain, or the control center of the cell. This is the nucleus, right? It controls all the activities of the cell, as well as it houses an amazing molecule called deoxyribonucleic acid, DNA. We have the cytoplasm, which is like a gel-like structure that holds the other organelles in place. And here too, we have several chemical reactions taking place. Next, the vacuole is that storage space in the cell. For plants, they have like a central vacuole. I'm going to show you. And then for animals, they can have many smaller vacuoles. Then we have another amazing organelle here called the mitochondrion. It provides energy for the cell to carry out its metabolic processes. Next, we have the chloroplast, only found in plants. Chloroplast, I just spoke about it during the process of photosynthesis, contains the pigment chlorophyll, which makes it green, and it is basically the photosynthetic machinery of the plant. Next, we have ribosomes. Ribosomes make proteins. Next, the endoplasmic reticulum. Mind you, you must be able to identify the two types of ER. There is the smooth endoplasmic reticulum and the rough endoplasmic reticulum. Why it's called the rough is because it has ribosomes on it. So therefore, the rough endoplasmic reticulum must be involved in the process of protein synthesis. Then we have the smooth endoplasmic reticulum that is involved in the synthesis of fats. Next, we have something called the Golgi apparatus, which is the factory of the cell. So whatever is made by the endoplasmic reticulum or other organelles, the Golgi apparatus is involved in packaging them, processing them, making them more suitable for their functions, and sending them to their destinations. Going to the exam knowing the organelles is not enough. You must be able to draw plant or animal cells. This is my drawing of a plant cell. I don't expect you to be perfect drawers, right? But you should know it, look in your head. So when you go on the exam, you can reproduce a drawing, right? And you should know where the organelles should be positioned, their names, and their annotations. Okay? This is my drawing of an animal cell. Here are some common organelles found in the animal cell. Again, you need to know the animal cell, look in your head so you can draw it when you go to the exam. Okay? And then you must be able to compare or contrast differences and similarities between plant and animal cells. Plant cells contain one large vacuole versus the animal cells. There are more than one vacuoles, but they're smaller. And then for a similarity, you could say that the vacuole in both plant and animal cells, they're used for storage. Remember, only plant cells contain a cell wall, which is made up of cellulose, which that's why it looks like this. That's why it looks like this, a rigid rectangular shape because of the presence of cellulose, okay? No cell wall for animal cells, but they contain a cell membrane, and that's why they are so irregularly shaped. You have some look like a circle, some look like a oval, or so forth. Remember, plant cells, they have the nucleus at the side of the cell. See there? And then for animal cells, they have the nucleus at the center. Another structure here only found in plant cells, chloroplast, right? The chloroplast is dispersed all over the cell, as you can see. The photosynthetic machinery, they're all over the cells. But in animal cells, they don't contain chloroplast because we are heterotrophs. That's right. We don't make our own food. We don't have time for that. We rely on plants to do that for us, thankfully. Lastly, you could talk about how starch granules are dispersed in the cell. Photosynthesis makes the sugars. The sugars are converted to starch, which are stored in granules in the cell. And then for animals, we don't have starch granules, but we have glycogen granules instead. Moving on to the next topic, this is osmosis, a process that occurs at the cell membrane, right? Remember, the cell membrane controls what goes in and out of the cell. And then osmosis is the movement of water molecules from a region where there is a high concentration of water to a region where there is a low concentration, right? So what is happening is that the water is moving from where there is a lot of it to where there are more solutes. And this occurs through a semi-permeable membrane. Osmosis depends on concentration. It does not require energy. And high-yield osmosis strategy is you want to go to the exam familiarizing yourself with at least 2 examples of osmosis in plants and 2 examples of osmosis in animals. Now, another term tied to osmosis is called tonicity, which is the ability of a solution to make water move in or out of a cell by osmosis. And I'm sure you've covered this in your laboratory exercises. We looked at what happens when cells are immersed in hypotonic, hypertonic, and isotonic solutions. So if we place a cell in an hypotonic solution, then water will move into the cell. Hypotonic means that there are more solutes inside the cell than outside of the cell. So that's going to make water follow that direction and move in. Hypertonic is the opposite. We have more solutes outside than inside. So water is going to move out of the cell. Isotonic, iso means equal. So there's equal amount of solutes on the inside and outside. So we're going to have no water movement. Lastly, I want to know how the plant or the animal cells look when they're placed in these solutions. Are they plasmolized, flaccid, turgid? You remember those vocabulary words? Yes, you need to revisit them. Right, moving on to the next topic, which is climate change. And come on, guys, from grade 6, we know the difference between weather and climate. We know that weather is immediate, climate is long-term weather changes. But what happens is that due to the anthropogenic activities in the environment, we have what is called climate change, which is a change in the long-term state of the atmosphere over multiple decades or longer. Now, what are the causes of climate change? The most important cause is the emission of greenhouse gases in the environment. And these greenhouse gases cause what we call the greenhouse effect. What are some of these greenhouse gases? Carbon dioxide, methane, nitrous oxide, soot, fluorinated gases. Those are some of the greenhouse gases that causes the greenhouse effect. Then some of you might be wondering, what is the greenhouse effect? So you see these same gases when humans carry out their activities, because we are the main perpetrators. When we carry out these activities and release these gases, They are going to trap the sun's heat, right? And this is going to make the earth warmer. And then due to the increase in the temperature of Earth over a long period of time, that is going to result into a process that we call global warming. Right, so what are some effects of climate change? Well, firstly, as I said before, Earth is going to be warmer, right? And then that's going to disrupt the functioning of many organisms that thrive in their optimum temperatures, right? There's going to be changes in the water cycle. There's going to be melting. We probably have more floodings, more tsunamis, and so forth. Lastly, we have current and future trends regarding to climate change. This is asking you to familiarize yourself with what is happening now regarding climate change and what scientists predict in the future. I'm going to post some links with some case studies and some scientific revelations as it regards to climate change in the description. Look in the description, there are useful links. All right, next topic, conservation of the environment. And we know that conservation of the environment has to do with these activities that we carry out to protect the environment from destruction, right? To promote sustainability so that we can have the same beautiful environmental conditions available for the next generations. We can do so by implementing laws to protect the environment, implementing sensitization programs to include education of the public on the benefits of protecting or conserving the environment. The government can implement monitoring strategies, to ensure that humans are actually conserving the environment. There are a series of agricultural activities called organic agriculture that we can do, again, here to conserve the environment. There is agroforestry, there is hydroponics, there is conservation tillage, right? I'm going to also post a link in my description for you guys to look on the various types of organic agriculture. Next topic is blood, right? What is blood? So we know that blood is a liquid connective tissue that has several important things suspended in it. What are these things? Cells, nutrients, hormones. What are some of the functions of blood? Well, blood helps to maintain homeostasis. Blood transport gases, waste, and nutrients. Blood helps us to seal injuries and fight infections. And when we look at these components of blood, you're going to see that they all work collectively to carry out these functions. The first component of blood here is red blood cells. Now, red blood cells, they are disc-shaped cells, red in color, because they contain hemoglobin, and hemoglobin helps to carry out their function, which is to transport oxygen and carbon dioxide around the body. They are short-lived because they lack a nucleus, and that's why they're so numerous too. On the other hand, white blood cells, they have a nucleus, right? And there are several types. There are phagocytes, lymphocytes. Holistically, they are all involved in the destruction of harmful microorganisms. They play a pivotal role in the functioning of our immune system. We have platelets, which are fragments of cells. And even though they are fragments of cells and they are not living, they perform an important function in the blood. After an injury, the process of clotting takes place, which is facilitated by platelets. You must go to the examination knowing the steps involved in clot formation. Remember, the platelets are going to release an enzyme which is going to convert prothrombin to thrombin, right? With the involvement of calcium. The thrombin is going to be involved in incorporation of a protein called fibrinogen to trap the platelets together, right, to form the blood clot. Sickle cell anemia, right? It occurs when the red blood cells become sickled or crescent shape. Remember, the normal shape for the red blood cells are biconcave, but in this disorder, they're sickled. And the drawback to this is that it's going to prevent the red blood cells from carrying out their major function, which is to carry oxygen around the body. So that's going to limit oxygen transport. All right. You want to know the various symptoms of sickle cell anemia and its effects. Remember, for the paper too, you don't have to study blood vessels and heart and pacemakers. If you were to do that, they would have placed the entire circulatory system, but they require you to just know the components or the structure and the function of blood. Moving on to the next topic, genetic diagrams. Sometimes the examiner might ask students to show how various characteristics of an organism are inherited, right? To do this, you must be able to use Punnett squares, pedigree charts. For your Punnett squares, you can use it to show the inheritance of one trait, and you'd just use a simple monohybrid cross. For the inheritance of more than one trait, you would use a dihybrid cross. I'm going to attach another link that is going to show you how to construct these charts because I don't have the time to work these out today. If you should use a pedigree chart to answer a question, you would use it if they ask you to show how a trait and the genes that control that trait are passed through a family. So I'm thinking that if you should get a hemophilia question, then a pedigree chart can show how that is inherited and how it is passed through the family. Lastly, we should know that the genotypic ratios are the ratios of the gene combinations in the offspring. And then for the phenotypic ratio, these are the ratios of the visible characteristics in the offspring. Moving on to the menstrual cycle. By now, we should know that the menstrual cycle occurs in women, right? And it is a cycle that shows a series of changes in the structure of the reproductive system to prepare a woman for pregnancy. What are the different processes or phases that occurs in the menstrual cycle? We have first here the follicular phase. As the name suggests, there is going to be a follicle being involved here. We know that a follicle is a growing egg. So this process is controlled by a hormone called the follicle stimulating hormone, and it is going to enable the growth and development of a follicle. After the follicular phase, we're going to have a process called ovulation, which is a release of that mature follicle from the ovary. All right, so after ovulation, we have the luteal phase. It is here that the corpus luteum, which is the ruptured structure that released the egg, it's not going to break down. What it's going to do instead, it's going to release progesterone, right? Progesterone is going to thicken the lining of the uterus, which prepares the woman for pregnancy. Important thing to note here is that estrogen and progesterone, remember, they're secreted. Estrogen was secreted in ovulation, progesterone was secreted in luteal phase. Those two hormones are going to stop the secretion of the first two hormones, which was luteinizing and follicle stimulating hormones, right? They do that because they want to prevent other follicles from developing. What happens if there is no sperm to fertilize the released egg? If there's no sperm to fertilize the egg, the corpus luteum will break down. Remember, it was the corpus luteum that secreted progesterone to maintain the lining of the uterus. Corpus luteum is going to break down and progesterone is going to stop. And then we can't have the endometrium being maintained anymore. So it's going to break down, and that occurs during menstruation. Right, moving on to the next topic, which is sexually transmitted infection. Right, so what is a sexually transmitted infection? So this is an infection that is inflicted or passed from the infected person to another through sexual activities. Right? What are the causes of sexually transmitted infections? By several pathogens to include bacteria, virus, fungi. What are some examples of sexually transmitted diseases? Well, the first one is HIV and AIDS, right? HIV stands for human immunodeficiency virus, and AIDS is acquired immune deficiency syndrome. To remember, AIDS is caused by HIV. Since it is caused by a virus, what would its treatment be? Right, its treatment would be an antiviral drug. So always remember, whatever the example of the sexually transmitted infection they give you, you must look at the type of pathogen that causes, because that's going to tell you the treatment. Another one is chlamydia, right? Chlamydia is caused by a bacterium. So the treatment of this would be an antibiotic. Similarly, gonorrhea is another sexually transmitted infection that is caused by a bacteria. The treatment here would be an antibacterial drug. Next, what are some effects of sexually transmitted infections? Well, untreated sexually transmitted infections can lead to serious health problems. Infertility, cancer, can also make a person more susceptible to contract HIV, and it can even lead to death. So guys, what I want you to do right here is spend a little more time on the examples of the STIs and their symptoms. We know how to treat them, we know how they're caused, we know their effects. Just spend a little time now on their symptoms. Now to the final topic of the paper 2, it is birth control. So here you're going to ask yourself, what is birth control? So by now you should know that birth control is a process by which men and women utilize several drugs or materials to prevent pregnancy. There are different types. You have surgical, barrier, hormonal, or natural methods. Let's start with the surgical, or better yet, sterilization methods. The first one is tubal ligation, right? Tubal ligation relates to the females. It occurs when the physician ties the fallopian tubes, so that an egg won't come down to the uterus and join with a sperm to create an organism. Tubal ligation is very effective. That's another thing I want you to focus on when studying birth control. Look at their effectiveness. Another thing to relate them to their prevention of STIs, right? So for the sterilization methods, they don't really protect a woman from or a man from STIs. You also have vasectomy. Vasectomy relates to the men. It occurs when the physician cut and seal the vas deferens. So I hope you can remember that. Vasectomy, vas deferens. Next, you have the hormonal, which is the utilization of hormones that we talk about in the menstrual cycle. Two main ones, estrogen, progesterone. The hormone methods include the birth control pill, transdermal patch, contraceptive ring, injection, intrauterine device, or intrauterine systems. Those are some examples found in the hormonal methods, right? You also have barrier methods of birth control, which is the use of a barrier, right? Something that prevents contact, like the condom that is placed over the penis, right? That prevents the entrance of sperm, right? For the barrier methods, they are normally effective, and an advantage to it is it protects the woman or the men from STIs. You have condom, femidom, which is a female condom. You have natural methods, right? You have abstinence or no sex. You have the withdrawal method. Some women, they study their menstrual cycle. It's called the rhythm method. All right, so what I need you to do here, go on it and start studying your hormonal, your barrier, and your natural methods. I gave you the names, so just read up more on them. All right, guys, thanks so much for watching this video. Next, you're going to continue watching and look at the questions that I've answered for you, past paper questions and my questions that I've constructed, remember though, I'm asking you, if there's 3,000, 4,000, 5,000 of you watching this, please hit the subscribe button. Don't be selfish. Share with your peers. Happy studying and good luck.